Greetings. My name is Mikayla Rodriguez, and I am a senior undergraduate chemistry and biology student at UT Permian Basin. Today, I will be discussing my undergraduate research project. In my research, I studied zinc oxide nanotechnology exponentials in the production of agricultural provisions. I was able to do so with the help and guidance of my research professor and mentor, Dr. Milka Montes, also receiving gracious financial support from the Welsh Foundation and undergraduate research program at UTPB. Nanotechnology refers to sophisticated technology in which materials we deem nanoparticles ranging from 1 to 100 nanometers in size are manipulated in such ways that they can carry out multiple functions in many fields such as biomedicine and drug delivery, cosmetics, textiles and fabrics, optics, energy, and in agriculture. In agriculture specifically, the most common uses of nanotechnology include controlled release of micronutrients, pesticide and fungicide properties, decreased loss of crops, and altered plant nutrition. The goal of this project was to observe the effects of a zinc nanoparticle fertilizer in comparison to a zinc nitrate fertilizer at varying concentrations. This was done against two basil plant varieties, sweet and Persian basil. The basil seeds we used for this project were purchased from Pine Tree Garden Seeds, and the zinc oxide nanoparticles were purchased from US Research in Nanomaterial Incorporated, based out of Houston, Texas. To enable efficient seed germination, we primed the seeds. To enable efficient seed germination, we first primed the seeds by soaking them in deionized water and stirring with the magnetic stir bar for about 15 minutes. The primed seeds were then left in a dark, cool place and observed until the seeds germinated into seedlings. 8 days after priming, the seedlings were transplanted to soil, placing 3 seedlings of pot per soil. Subsequently, the plants were maintained at 65% relative humidity, 20 to 25 degrees Celsius, and a 16 8-hour light photo period while they matured. During this time, fertilizer suspensions were prepared at various concentrations for both the zinc oxide nanoparticle solution and the zinc nitrate solution. For each type of fertilizer, suspensions at 0, 0.5, 1.0, and 1.5 ppm were prepared. To ensure adequate homogenization of the suspensions, they were placed in an ultrasonication bath for 1 hour at a frequency of 1200 kilohertz at room temperature. Presently, there exists multiple methods of applying nanoparticle-containing solutions to plants. In this particular experiment, the foliar application method was chosen. Each fertilizer suspension was placed in a sterile plastic bottle in 10 milliliter aliquots. These fertilizer suspensions were stored away from direct light as to avoid the photoreactivity of the zinc oxide nanoparticles. 72 days after the seeds were transplanted to soil, the fertilizer treatments began. The plants received a total of 4 2.5 milliliter treatments 4 days apart. In total, each plant sample received 10 milliliters total in the duration of 16 days. Samples of sweet and Persian basil were treated against the 2 fertilizers at each of the 4 concentrations. Throughout the duration of the treatment, plants were regularly observed for any visible changes. Observations of the impacts of the foliar spray fertilizers on the plant species. Observations of the impacts of the foliar spray fertilizers on the basil plants include the following. Plant samples treated at concentrations higher than 0.5 ppm appeared to experience discoloration in both the zinc oxide nanoparticle solution and the zinc nitrate solution. Interestingly, the discoloration occurred Interestingly, the discoloration occurred more frequently in the plant samples that were treated with zinc nitrate, which can be found in most widely used fertilizer sprays. In addition, the plant samples at concentrations 1 and 1.5 ppm seem to grow at a decreased rate than those at 0 and 0.5 ppm. This was largely observed in the shoot region of the plants at the end of the 16-day treatment. The preliminary findings of this project offer evidence suggesting that perhaps nanofertilizers applied via the foliar route may have less harmful effects on plants than other widely used fertilizer sprays containing zinc nitrate. Consequently, further exploration of nanotechnology applications in agriculture could lead to more efficient and environmentally friendly practices of growing crops. In conclusion, the findings of this project suggest that at concentrations lower than 0.5 ppm, nanofertilizers may be more positively correlated to plant growth and development than other widely used fertilizer solutions. When treated at concentrations higher than 0.5 ppm, the plants were observed to have discoloration of leaves as well as decreased rate of growth. It is important to note that the previously mentioned observations occurred less frequently in the zinc oxide nanofertilizer spray. In the upcoming months, more detailed and specific evaluation of the samples will investigate the biochemical constituents of the basil plants. This project received funding from the esteemed Welsh Foundation as well as the undergraduate research program at UT Permian Basin. I would like to give special thanks to my research professor and mentor, Dr. Mikayla, as well as the Department of Chemistry at UTPB for the continued support and guidance.